Hi, I'm Oliver.
And I'm Ben.
And welcome back to Orange. Today we are interviewing a bioengineer from Monash University who's passionate about improving mRNA therapy.
You recently developed the first mRNA therapeutic program in Australia and recently was awarded an ARC Early Career Industry Fellowship to refine the quality of next-generation mRNA vaccines.
Welcoming to Orange Podcast, Harry.
Well, hello everyone. I appreciate Ollie and Ben for inviting me. So my name is Harry. I'm a bioengineer based at Monash, and it's been great to know you guys and to come and join your podcast. Really looking forward to have a chat with you today.
Absolutely. So Harry, you've got a lot of experience in the mRNA space. Just straight off the bat, for those in the viewership who don't know what mRNA vaccines and mRNA research is, can we just have a bit of a lay audience description of the science?
Right. So mRNA is a messenger. It stands for messenger ribonucleic acids. And what you really need to know about that is mRNA is a molecule that found in all our cells. And its goal really is to communicate between, the DNA inside our cells and the function of the cells. So it will tell the cells to do something, move this part or, do this type of function, which really helps organize our lives and how we live inside the cells. So the way that we would use the mRNA as a vaccine is we will be able to code for the antigen that we want to do and we want to vaccinate against, and put that within the messenger RNA, formulate it, and then send it inside the bodies. Then the cells some cells, specialized cells, these are immune cells, would pick up that vaccine, make it in the way that is being made like the viruses for a very short period of time, perhaps a couple of days, just enough to help train the immune system to recognize this, antigen and create the necessary reaction. So then it will protect us if that virus came along. So in a way, it's really a code of life. It's so central to biology because DNA, think about it, is a storage of information. You could probably, add more to it, but it's actually adds the function, what interprets that storage of information from the DNA to whatever function is. That's the mRNA.
Precisely. So, I've had the, opportunity of teaching my younger sister some basic science, for example, and I always like to use analogies. And I feel like a really good analogy to describe mRNAs in general or even DNA is almost like the library. Like you walk into a library and there's a lot of books everywhere,
right? But
it's not necessarily specific. And then when you get to the actual book, you open it and that's the mRNA. You're learning information and that's changing your behavior. They're very similar to how mRNA would change the behavior of a cell.
Yeah.
So
exactly. It is basically the transcript, the message to which are being copied from the library, from the DNA. So, I love that metaphor.
Now, you've been involved in mRNA research for a while before the mRNA vaccines came out. Obviously, this wasn't in the public zeitgeist a few years ago, but then it rapidly became into the public zeitgeist. What about mRNA technology made you so enthusiastic about it that made you want to devote your research career to it?
Yeah, and I think that's, reflecting back on that now and some time ago, perhaps I should reflect a bit more to bring all these kind of elements together. But actually, in my undergraduate degree, I took an elective I did a pharmacy degree at that time. But I took an elective called biotechnology, and at the time, what I studied, they did not have biotechnology kind of within the main kind of main course. But I did take that, and really fascinated me that some viruses is being trialed to, and some DNA viruses, someone called adenovirus is being trialed for gene therapies. I absolutely love that. I thought that is an amazing thing. I started learning about nucleic acids, and fast forward, I went to do when I came here to Australia. I wanted to do kind of study that phenomena, that delivery phenomena. How can we deliver DNA to the cells, and it is still to this day one of the biggest problems in medicines. And there's an improvement on that. There's now therapeutics, but it's still something that if you can really utilize, it's amazing. But it is very, very difficult. And I spent throughout my PhD studying a way to try to mimic viruses, because we don't want to keep using viruses, especially for gene therapies. You need a large dose, but quite complicated. So can we do that without the viruses? Can we mimic a way to do that without viruses? And that was my PhD. But we really, in a way, as a lot of the PhDs go, and you guys would know that as well, things don't always work out the way you think they would. And that was a particularly difficult problem to really tackle. So in about 2017, we kind of been looking at what can we do, what sort of new technologies we can adopt. And one of them was there's a rise in the use of mRNA. That was actually before using lipid nanoparticles and mRNA. There was some liposomes, which are the early version, very kind of very basic versions of lipid nanoparticle, very early versions. But they've been using mRNA, and when we looked at it, it's been kind of, there's some improvements have been happening consistently in the past few years, and we thought that there's potential that we kind of get improved for a few years, it will actually work. So we'll get to a point where it will work. And that's where the decision was made. Well, if we're not really failing with the DNA, can we use mRNA to do that? And that was a decision in 2017 when we, Colin Poughton and I, who's at the time was my supervisor, decided, okay, this is what we want to do. So I finished my PhD around 2017, 2018. Actually, end of 2018 when I really finished and submitted. But we already had started working on mRNA. And then after that, we kind of kept going, working on it. So, and we did really, we had a really good advances before COVID-19. And when COVID-19 came along, it was kind of the obvious thing to do. We have not only worked on, but we developed our own formulation technologies. Had two formulations. One of them was based on lipid nanoparticle that showed improved uptake to those immune cells that I was telling you about, which were really important to make a vaccine successful. So we already had that technology ready. It was needed to be improved a little bit, but it was almost there. We already had all the parts of the RNA, a lot of IP already ready to go. So we were in a kind of very good position to really to take that on, and we did.
Fascinating. I feel like the timeline of when you pursued your PhD research project and then COVID-19 came along was quite serendipitous. And how did you feel when the pandemic kind of became publicly known? Did you come to the realization and think to yourself, this may be an opportunity to use this technology? Or did you think that was many more years away?
Yeah, look, I think, so it was a kind of an immediate realization that this technology would work really well. No one would work well, actually, I would say, but no one kind of anticipated it's going to work that well. But in a way, you're right about there is serendipity to it. And I think I'll tell you a little bit more about what I also think is important, to everyone maybe that's listening or interested in pursuing, what sort of subject that I should pursue. But I think it's not only serendipitous to us, it's to everyone in the field, because at the time Moderna was struggling, BioNTech was pursuing their cancer. I think they were going well at that point anyway. But there wasn't a lot of money in the mRNA. There was some money, pharma has been paying attention. What happened, I actually got invited by by a committee who was headed by the now Nobel Prize winner, Kathleen Carricker, in 2019 to the mRNA Health Conference to present my work about spleen delivery. And at that conference, so that conference organized by BioNTech, Moderna, and CureVac, and they invited me to come and give a talk. And among the talks, though, there was, with our also work, but all the other work that has been done on the vaccines, was kind of a quick realization that this technology cannot work. There's some really major improvement that happened between 2017 and 2019. And that was the inclusion of lipid nanoparticles, and really the kind of the ultimate realization that this is, we're going to use that. We're going to really try to optimize it towards that. So the formulation being optimized, how is it get formulated, the efficacy of the mRNA got boosted phenomenally for the vaccine use. So at 2019, it would have, we knew it's going to work in a way. We just didn't know how long. But, Colin and I went to that meeting in Berlin, and I've written about it actually it was November. I written about it in November 2019. That was a month before. So you can find it online. I wrote a blog about it. That was a month before the pandemic came along. And I said, this is going to be important for vaccination. We need to build a manufacturing capacity in Australia, which we don't. And I can come to that later. But I've kind of talked all about it, that you can hear about these things now. But I think it was really we realized that this is going to work. Which we didn't know it was going to be 2, 3, 4, or 5 years, but we know the technology is going to work. We still didn't expect how good it was going to be. But about in February 2020, I sent an email, and I was asked to get some of those historical things because people in the it was actually the Australian wanted some kind of backup story about how it's been developed, the vaccine been developed. But I looked into it the other day, and it was me sending Colin kind of like a very short, I think it was like 4 words. This is important. We should do something about it. So that was about very early February, and he said, yes, I agree. And what I did straight away is I've just sent the sequences to be manufactured in a way that we can make an mRNA out of them. So I've sent 4 designs, and that was before March 2020. So that was very early on, and by April we had the vaccine. Before April, we vaccinated, and by May we had the Australian make a story about it. So we were ready. We were actually ready, but obviously no one came to fund at that time because everyone was betting on the protein vaccines that we would develop here and AstraZeneca. And even through that, a lot of people criticized the mRNA. But that obviously turned out to be the one that worked really well and is the right technology for this sort of thing. Yeah, not to take away from the proteins, but they also work. But yeah,
in Berlin, or after you came back from the conference in Berlin, you wrote, "I must write about mRNA therapy so more people can know about it, because when mRNA therapy reaches its age, it will touch most of us."
Yeah,
and I think that's just very
predictive.
Very
prophetic.
foreboding and prophetic. Yeah.
Yeah, it's because I still believe it. I think actually I believe it not only more now. I believe it's far, far broader. And I tell you a little bit more about that, but later. But I think it's going to be more than just health. I think that's very important because I think every part of synthetic biology, because it's a code, the messenger code. It's the way that cells, even the bacteria, how the bacteria even how viruses communicate.
Well,
it's the code of life.
Yeah, let's talk about it now.
Yeah.
Let's talk about it now. So are you suggesting that there's future utilities beyond vaccines for mRNA?
I think it's not only not only that, there is obviously, I think now beyond doubt that there is a lot of applications beyond the vaccines with using mRNA. So we kind of seeing a lot of potential use for cancer therapeutics, and that's sometimes called cancer vaccines, but in fact it's almost like a therapy. It's called therapeutic vaccine. So you give it after the cancer, or even sometimes in some rare cancer you give it before the cancer, just got anticipation for that. So we call it prophylactic, so protective in a way before it happened. So that, I think, that's very well advanced. There's a lot of logistical issues with that, but I think that is a very interesting case in the mRNA because that relies on what we call personalized medicine. It's a new philosophical concept that, because each cancer from each patient is different, then you will end up having to vaccinate against that specific cancer. And to do that for thousands of people is phenomenally logistically problematic.
And expensive.
It's very expensive. So our infrastructure has not been made to, work on mass customization. We can make one mRNA, millions of grams of it. That's fine. We can do that. But we can't make million different mRNA at 1 milligram of. So it's a very, very logistically difficult. It's like the same thing happened in 3D printing, where you have this approach of mass customization versus mass production. So this is an interesting problem. So this is kind of where you have the mRNA facing both challenges and great opportunity. Then you have, the genetic, solving genetic diseases. I think that's going to be phenomenal. We're already working on that. We have a big program on that with Children's Medical Research Institute to try to really target almost now all the potentially this technology we're developing, all the congenital liver diseases that kids would have. So it will correct and add any new therapeutic that you want to really, cure these kids, regardless basically of the disease. And it's just an incredible thing to be able to do. So these kids would have a kind of a missing enzyme when they were born. So we could basically replace that enzyme. We can correct it, but we can also replace that enzyme by inserting a new type or kind of a new functional copy of that enzyme. So, those kids will recover. So it's a massive opportunity. And I think we're almost there. It's just a kind of a dose problem. So that going to come along. And that actually is combined with another technology, which you probably heard about, and you guys talk about, called CRISPR-Cas9. So, CRISPR-Cas9 combines all these protein gene editing, and mRNA is very well suited for that. You have, the Cas9 already, CRISPR system already have an RNA components, but we're also working on kind of how can we make the Cas9 and other versions of it using mRNA, and how can we organize that into completely non-protein, non-viral system. And that's a very important part to that kind of whole story. So that's the therapeutics, and I can probably go for hours about what is potentially, but I also think that it will really impact synthetic biology a lot, particularly, using bacteria for the environment, producing meat, things like that, animal health, crop engineering, because all of that brings it, you're not editing the genome. It's not a GMO. So all of that, both regulatory and safety, is far kind of more appealing and more, have a lot more impact. So you could do that with mRNA, but you need a lot of it.
I have a brief question, just on this note, is like, out of all the kind of research that you know of in the mRNA space, what do you think is the most fascinating project that you've come across? One where you look at it and you just think, wow, that's absolutely incredible.
Yeah, I think I do get mesmerized a lot. So it's hard to pin it down to one thing, but I would say what is happening in gene editing and what we call gene insertion, I think it's phenomenal. It's the sort of things that will do. It's you're talking about, as a technology, so early. You can see in animal models now some really clear curative responses. Almost, if you think about it, you can solve almost everything as long as you can access it. So the problem is actually the access of it. So that's what really, I think that's an amazing, potential for mRNA. It is definitely going to be the second most important application after vaccines. And I say by important, I mean in terms of being ready. So this is going to be, it's already kind of almost ready technology. So gene editing, gene insertion using mRNA, you're still going to use the Cas9 technology, but the packaging and the drug becoming a drug is much easier to happen on the mRNA. The other thing is, I think, the potential of using mRNA for, again, personalized medicine. I think that's an amazing, both from to cure specific patients, specific diseases that only one patient would have. So gene mutation that you can correct, but also for, cancer. So if you have a cancer that is specific for that person, can you vaccinate against it? Can you give a therapy that work only for that person, but it's well tailored specifically against that cancer? So you'll have the maximum response against it. I think that's a phenomenal thing as well.
So let's go back to the core concepts of mRNA therapeutics. Most people are aware of, because of the pandemic, are aware of using mRNA to code for a peptide or part of a protein of a virus. So you put the mRNA, you package it into a lipid nanoparticle, you give it to the patient. That lipid nanoparticle travels to a certain cell type in the body. It actually enters into the cell, it enters the nucleus, and then is transcribed. Doesn't enter the nucleus, but
No, it doesn't.
It doesn't enter the nucleus. It is
We
transcribed.
need to emphasize on that. Yeah.
It's translated into the protein, and then we mount an immune response against the protein.
Right.
You've also described mRNA that is used to replace a certain protein or enzyme that a patient doesn't have. And then you've mentioned like cancer vaccines and therapeutics. I guess just for the audience's sake, how is a cancer vaccine and therapeutic different? What are we actually coding for? Are we replacing something? Are we adding something?
Right. Yeah, so it's the therapeutic cancer vaccines work in a way very similarly to what COVID vaccine would work. And I would say about that is that if you replace the thinking of it's an antigen from a virus to an antigen from cancer that is only being made in cancer. And the reason for that is when the cancer divide and multiply and in fact causes a lot of mutagenesis, some of these proteins that get mutated and open up during the cancer growth, these are very specific to the cancer. They're not really mutation that you can find in the normal cells that surround the cancer. So when you vaccinate against those very, very specific mutations, you could mount an immune response against the cancer only without the cells. So, in some senses, that does work. And you really need to find the right antigens from those cancers. So we call them, the scientific term called neoepitope vaccines. So they are, they're small bits and pieces that are found only in the cancer, but not found in the healthy cells. And you would vaccinate against them. And so you put them into the mRNA, those antigens. You would deliver it. The specific immune system will pick up the mRNA, will take it into the cytoplasm, will make it into an antigen, will process it, will present it to all its friends that are immune cells that are really interested in looking at it and creating an immune response. And then those cells will then go around and search for the cancer and then interact with the cancer and kill it. So, it happens not always, because you really need a very strong immune response to counteract the cancer. But it happens in enough patients that it warrants an interesting technology that we want to pursue.
So I guess I'm curious about this because usually when we create vaccines, we do it for a antigen that the patient's immune system would ordinarily mount a response to, but we want to give them sort of immune memory so that they're prepared to encounter that specific virus or pathogen.
Right.
But with a cancer vaccine, the antigen that's in the cancer, the patient doesn't ordinarily mount an immune response to, potentially because it's sort of a self-antigen. We have immune tolerance, centrally. So what is it about a cancer vaccine that, I guess, forces the immune system to mount an immune response to what it would ordinarily never mount an immune response to?
Yeah, so that is right. And I think the word you said, tolerance. So we have, of course, a lot of tolerance for some of the antigens that are found in the cancer. And one way to do that is to at least in part break that tolerance and really go from, so you need to be obviously careful about which protein you select, what protein you don't vaccinate against. But also, it's not just that. It's a lot of these selected type antigens or proteins. So antigen is a bit of protein. That antigen or that epitope is actually a mutant. So it's not really what we found in our cells, which actually that makes the immune system, because our immune system is very, very sensitive to even one mutation in the protein, it responds very differently. So it will recognize that mutation and will counteract and kill only the cells that have that mutation. So that's how the neoepitopes works. There's other kind of versions of the cancer vaccines. So some of them called, trying to remember on top of my head, but they're tumor-associated antigens. So these are antigens that normally they're not found in the cells, or very, very low, but they're found in cancer cells and be made in high quantities, and these could be counteracted against. The important part about the personalized cancer vaccines or therapeutic cancer vaccines is that you need to take the cancer and sequence it, and really take that, process it and sequence it, and then design your antigens accordingly. So it's not a pre-designed, a lot of the neoepitope vaccines in particular, it's not a really, it's not a pre-designed product. It's a neo, so it's a new antigens that has been kind of emerged in the cancer.
Harry, I want to talk a little bit about the skepticism in the field of like mRNA research, because obviously since the pandemic, a lot of people have become, skeptical of getting the vaccine. People are skeptical of the technology, how safe the technology is. Can you talk at all on that note?
Yeah, absolutely. And I think, as you guys already probably know, any impressive technology brings kind of a lot of skepticism and fear because, I think it counteracts, I think it's a natural human response to enthusiasm. So you'll end up with a lot of people enthusiastic about technologies, if you think about AI, and then especially with the recent drama, and then you have people who are just probably from a natural balance of society would be very skeptical about it. So that the more the technology, the more advanced or has potential, the more there's skepticism to it. I think a lot of the skepticism was unfortunately political. These mRNAs are incredibly safe products. They are without a doubt, one of the safest, thing you can think about, especially vaccine. The level of how much they protect us versus the reactive response to them is really good. But you want to always think about, any drug, any vaccine is you'll always have some side effects. So maybe a little bit of pain in your arm and something like that. But I think a lot of that is really not. I don't think the conversation has been about the real side effect. It's about some imagined side effect that is being driven by anti-vaxxers or politically motivated. I think when you look at that, it's really loud voices, but when you look at the number, it's not really that concerning. However, a lot of that has been tied, all of the vaccine has been tied to other things like, lockdowns or, the other kind of political things even in the US. And I think that's what caused a lot of issues as well, where more and more people became skeptic. Yeah, I don't think necessarily, I think when people get to learn a lot more about it, it's gonna be, they'll be more accepting. Also, one more thing about that, I think for therapeutic perspective, you don't have to necessarily worry about that too much at all, because it's a very different equation between a healthy person getting a vaccine, or especially if they're forced to take a vaccine, versus
cancer patient or something.
A therapeutics where it's a response to something. It's not affecting the freedom. This is a complex conversation, and I know people has been really pursuing a lot of that, the background that I know of, and I think it's a really fascinating research. But I think it's in terms of the numbers, at least in Australia, is not that concerning.
Yeah. Great.
I'd like to come back to the sort of serendipity point that we were discussing earlier. I feel like a lot of the most successful people in the world, they picked a field and they pursued it because of this sort of innate sense that they really loved it, or they thought it was the future. And then they pursued it at the right time, right? So Ilya Sutskever pursued AI and he joined OpenAI right at the time when these large language models are going to be scaled to such impressive performance. And then you have pursued mRNA vaccine technology, or just mRNA technology more broadly, just before the pandemic, just before it became commercialized. But then a lot of people also struggle to retrospectively work out how to identify those opportunities. How do you know what the future is so that you position yourself for that future? And maybe you have some ideas on that front.
Yeah, look, I think, you want, I think that's for obvious reasons, and we're smiling now. It's a very complicated kind of question and answer. And in fact, I think part of it is you need to be, it's mortifying, but you need to be ready to be wrong and to keep going. I thought, the DNA going to work, I was wrong about it. I actually have developed something that I would really, aside from all the things that I should have developed, but one thing I did develop a long time ago was it's actually, I used to not journal, but just put in a sheet what I think of a new technology come along or is coming along, what I think is going to work or not work, and why do I think that. And I leave it, I'll come back to it 2 years, 5 years, 10 years later. And I not only check if it works or not, or I'm right or not, whether the reasons are right or wrong. It's very hard. You have to really have a comprehensive understanding of the field. I think you need to be, in a way, somehow generalist to start thinking about what sort of next technology going to be good. Because you really need to, most of the technologies emerge from tying up knots. We really don't, I think we run out of potentially kind of one discovery that, that you can take it so deep, you can improve it. It's more, they're becoming more and more difficult. They're rarer to get by. So, discovering something about, you discover something about quantum mechanics that leads you to build a computer, or something like about semiconducting that gets you to build a transistor. I think they're becoming more and more rare than that. So a lot of what you could do is to try to tie different components of technology and think how they're going to potentially fit together. And you imagine it. You just say, okay, that's how I think the future is going to become, because that's how I wanted it to become. And if you're smart and someone else is smart, probably as smart as you're smarter, then you guys are going to reach the same conclusion. So it's really that. It's like almost, there's an eventuality, I think, to technology that we don't talk too much about. It's, if I don't do the technology that I did, someone else would have done it.
Yeah. Do you have any examples of something that you may have written down that came into fruition? Yeah,
I think sRNA. I think I nailed the sRNA thing. I thought that sRNA, actually, I think I nailed it twice, but I felt that the AAV, I did not get that one. I thought it was going to work a lot better than it worked now. AAV vectors and using AAV for gene therapy.
Like adenovirus.
It did work.
Vectors,
AAVs are adeno-associated viruses. They're kind of a new, updated, a lot safer modality that is used for gene therapies. And I thought that AAV is going to be far, far more advanced now in 2023 than I thought in 2016. And I was still right about the trajectory in 2017, 2018, but then, because there was drug got approved, but then they kind of stopped falling off, and they're really becoming more and more difficult to develop. There's a lot of reasons for that. sRNA, on the other hand, I think I got right in some way. I thought that the lipid nanoparticles that we actually now use in the vaccines will not be the primary vehicle for delivery of sRNA. And I think that kind of now became the case. So the sRNA, the first drug ever used for siRNA, which stands for trying to remember the full thing, but it's basically silence genes. So it was short interference RNA. But SI is kind of, in a nice way, it also stands for silencing, and the actual phenomena called RNA silencing. So it actually it does the opposite of the mRNA. It shuts down an mRNA that's found in your cell. So mRNA that's too much of it or harmful or affecting, causing a disease, so you can shut it down. So it's also kind of the opposite of mRNA. And that was developed, actually the whole lipid nanoparticle system was developed for sRNA, was developed to deliver the sRNA to the liver. And there was a drug called Onpattro that was approved in 2018. And beyond that, there was to be the first and the last lipid nanoparticle sRNA product. I don't think they're going to abandon it as well. It worked, but they've managed to develop the technology on the sRNA a different way that then will become that doesn't need the lipid nanoparticle. So these are competing theories about in 2014, and we just weren't sure. And actually, the lipid nanoparticle took over, and they were the successful. They got there first, but as the other technology developed, then that took over, and it's become the norm now.
Just for the audience's sake, there are therapeutics and there are delivery mechanisms for the therapeutics. So in terms of the therapeutics, we have the mRNA, we have CRISPR, we might have mRNA for CRISPR, and then we also have the delivery vectors. So we have viral vectors where you put the stuff in the viruses. We have lipid nanoparticle vectors where you put it in a lipid nanoparticle, basically just a fat globule.
Yeah.
And then the sort of other, I guess, delivery mechanism, and you can touch on others, but the other one is independent of anything, right? So you deliver it by itself. Is that right?
Yeah.
Can you explain, like, my understanding is that there has been a lot of recent developments in LNP independent delivery of, CRISPR, of mRNA, of siRNA.
Yeah.
Can you explain that a little bit?
Yeah, absolutely. And I think so when you look at sRNA, for example, the other that I was talking about, the other competing technology called GalNAc, it's basically, think about it's a branch at the end of that branch. It's a branch that stands on the RNA molecule at the end of it. And that branch kind of it's like fingers, your fingers, 3 fingers. And these 3 fingers will have molecules at the tip of their finger, and that will actually go in and lock with a specific receptor in the liver. So a receptor which take up naturally these, if you think, well, for the sake of the analogy, take up these fingers. They naturally designed to take up those fingers. So that's how they do that. They piggyback on that mechanism that's found already in the liver. And that's technology called GalNAc that doesn't need a lipid nanoparticle. You can attach that to the sRNA. But the problem was early on that the chemistry of the sRNA originally was not enough, was not sufficient to overcome the natural enzymes in our blood and system that are programmed to destroy RNA. If you're an RNA molecule, the most hostile environment is, the stomach and the blood. There's so many enzymes that want to destroy you. It's because the body obviously thinks that RNA viruses, their place shouldn't be outside the cytoplasm. All mRNA of the cells is inside the cytoplasm. So the cells have developed, and our body developed a sophisticated system, which we call RNases. And these, like scissors, they just cut all types of RNA. So for sRNA, they've improved the chemistry so much that you've overcome the ability of the RNA enzyme to stop it. And that's the sRNA. That hasn't happened to the mRNA. It's quite more difficult, more complicated, because some of these modification it will just turn the mRNA doesn't work. So your ribosome wouldn't recognize it. So I think it depends on that. It depends on what sort of technology. I think there's a lot to be done more on lipid nanoparticle free delivery. I think that's very interesting. If we can do that, that will be great. But so far, obviously, just to come back to your question, is that we actually did solve the problem of treatment of genetic diseases, of vaccines in a way. The only problem that we have is delivery now. So delivery has always been, because again, it's a code to get whatever therapeutic you want. We already know what code we want to use. We can optimize, we can improve it, that's fine. We already know the code. If you're missing a protein, we already know what protein you're missing. We're going to code it, and if we put it on cells, it's going to work, right? But to put it in your body, it's quite a very difficult thing to do, and especially to deliver it in your body. For example, we have collaborators. We're working on all these editing, gene modifications, and using mRNA and the CRISPR technologies. But we're not delivering it inside. We're actually using what we call ex vivo. So ex vivo is you bring cells, CAR T cells or other cells that you can extract from the people. You put it in a dish, you add those elements, and you put it back in. And that is amazing because you could really you have the cells right in front of you, you would deliver them to the cells, correct the cells, do the function you want, and put them back into patients. And obviously that you can't do that with the liver because liver is an organ, but you can do that with any blood cells that you think about, all the immune cells or the blood marrow cells, and all. So you can think about solving all these genetic diseases. And actually, that is, again, this is quite straightforward. I think that will work spectacular. We're going to see a lot of new genetic, new therapeutics around that because it's so easy to deliver. So all of it is just the things we're seeing right now in the animal models with our collaborators is just blowing our minds because you look at it and you're like, okay, this is just cured it. That's it. It's just done. It's just these mice are very healthy. They're fine. It's all good. It's all worked.
Yeah.
It's just an incredible. It just works so much better than you think you need to work. You just actually now almost you have a big window to which you're not just touching what you need to the therapy, which we've done in pharmaceutical development, almost just trying to solve a disease. But now we're like way above what we actually need. So now we just scale back if we want to. I think that's amazing because that really brings this technology to fruition. I think that's going to be, a lot of the blood diseases is just going to be solved. That's how incredible is that?
Yeah, it is, absolutely. It's fascinating.
Within reach.
Yeah. So I suppose like an issue that there might always be in this case is accessibility of the technology.
Right.
Because it does fall into the category of personalized medicine, right? So in that case, how do you think that that's overcome in the future?
Yeah, and
do you think there'll be almost like wellness centers?
Absolutely. I actually do think that. I think you've nailed it. I know you're laughing now, but I think it's probably one way to do this is to have a specialized centers to which these treatments can be administered. I think that's how have a lot of impact in health economics as well. We can talk about that for maybe hours, but I think doing that within a hospital is a very difficult thing to do. Although it will be done, and I think I do love that happening, but I don't think it's the only place that this is going to happen. You'll have a kind of a specialized super clinics, maybe attached to hospitals, that's all fine, but they're independent, they do that. But I think what you touched is very important because I'm very passionate about that. This is what I want to help improve from a technological perspective, not from a health tech or from an economic perspective. But I think you talked about technology equity and how can we empower and enable this technology to become a truly personalized, championing personalized medicine, but also doing it at a cost both that beneficial for developers to develop the drugs, but also for patients and for our government to pay for it. Because I think that's so important for a lot of patients to have that ability. And that's why I'm working on manufacturing technology. So I've actually shifted. So my initial kind of the IP and the patents that I developed and the inventions were around RNA therapeutics and RNA delivery. But I start realizing that we're developing these amazing medicines, but we're actually not going to be able to deploy them to people unless we solve the manufacturing. It's like hand in hand. You need both to drive one over the other. And I think that right now, and not only manufacturing in a localized fashion, manufacturing at a higher quality, producing that manufacturing in a customized way. So the cost of one patient is probably similar to the cost of 100 patients. So we really have a lot of challenges to get there. And I think that because if you build these technologies, manufacturing technologies, that has an impact not only on one disease, but on all these other diseases that this technology will develop. So it'll have a kind of an outclassed impact to it. So I'm very passionate about that.
Yeah,
so
I can tell.
You can probably see from my face. And that's what I'm doing right now. It's my mission. It's my focus on. Yeah.
Absolutely. So you mentioned before we jumped into the podcast that you were kind of ruminating over transitioning out of academia and more into potentially like the startup world. Is that right?
Yeah, that's right, and it's probably it's the worst kept secret now, but everyone probably knows about it. I mean, I think we definitely are very, very not only passionate about this but committed, and I think the reason for that is to achieve that mission for to enable these technologies to be deployed and to empower people, scientists or doctors, to really treat people in that way. And across all the world, you need to really develop the technologies to do that, and we can't do that in academia. It's not just academia and research institutes are not programmed to deploy that at a larger scale, and we need to do it. We need to do it now because it will impact a lot of people. So I will be ready to talk a lot more about that. You will start looking at once the the 2024 comes along. So starting January, you will hear a lot more about it, about the name of the the startup we want to go for, and what we want to do. So what we're doing now is we're building the technology. We're going into a cave. Brilliant. Just building day in, day out.
Second interview on the horizon.
Yes, happy to. Yeah, so we'll definitely hear a lot more about it. We'll do a lot of media pitches, things like that.
Awesome.
Next year.
And that's really exciting. I'm very happy for you, and I think it's very much needed. It reminds me a lot about, you remind me in a way kind of like the Elon Musk of biotech and mRNA, because it's not just the machine that you produce, it's the machine that builds the machine. That is what creates economies of scale that allows you to actually turn what you've created into something that people can actually use and buy. And you mentioned an important point about the cost of this, right? Like you said that the current, manufacturing processes for mRNA are very capable of mass producing a single mRNA, but they're not very capable of producing, hundreds of thousands of unique individual mRNAs.
Right.
Can you talk about that a little bit, and where do we go from here? How do we turn what we have at the moment into something that's commercial and personalizable?
Right. Yeah. That's an incredible. That's a great question, because I think there is a similarity to see that and to imagine it, how it's going to work. And that's if you look at the revolution that happened in 3D printing. So 3D printing cannot compete with mass production, no matter how efficient you have. So when you hit something around 10,000 and more, you're really not competing. You can't compete with mass production. But if you're trying to develop, 1,000 or 50, or 1,000 different parts, then that where it becomes really when your 3D printing revolution would work. And even like a professional 3D printing is what you want to do. And you can manufacture even, things manufactured at a smaller number. So that's how you want to think about it. If you're not going to think about, so the medicines that I think in 20 years, for example, a lot of the medicine is going to be made, in-house in these wellness centers or in hospitals, and these will then given to patients on the spot. A lot of them is going to be personalized. If they need to be, if they're mass produced, then that's probably not the way you do them. But we're talking about genetic diseases and the host of those, vaccines, specialized treatments. So you'll think about that. That's going to be all of it within a small environment. But I think it also goes beyond that. You're touching about discovery research. All of this needs customized solution, rapid development, rapid iteration. You need to iterate. So we had to develop the vaccine. We had to iterate it a few times. Every time that's going to take you 3-4 months, you're just waiting. You're not doing any work. But doing that, if you're being able to customize and build machines that can build these machines at a very not only fast rate but also in a cheaper fashion, you will first have more people access the technology, and therefore the collective creativity will improve, and therefore more medicine will be developed. So why we always say, what I want to say about, keep saying empowering scientists and doctors, because you give these people the tool and they will create and amazing things. So you build a machine for them to build the tool, to build the whatever therapeutics you need to do. And the second thing, which I think is important, I think actually doing that, even doing it cheaper, will create more money. And I think people don't realize that. People who are offering the kind of the really, there's no real solution right now to this that I can think about, but adjacent solution has been offered in the market right now. They don't like that because, you know, they think that it's not going to bring these equipment that they provide, these tools cost a lot of money. They're really junky. They really don't work very well. But I think actually that if you create something that's much, and you create it at a much cheaper price, you'll have more people access the technology. So you're actually ending up having more money because it's an economy of scale in a way. More people having access to it, more therapeutics being distributed across the world. If you make something with like 50, 60,000, you're not talking about just the US. 50,000 is not much for a therapeutics, for 50,000 for the machine, but the therapeutics will cost a couple of thousand. That's not a lot for most of the Middle East, for most of the Eastern countries, so European, and now African countries as well. You know, it's not a lot. So making things more accessible, that's both price and both in terms of the technology equity, I think that's going to be very important. And I think, you know, people who will do that can actually make money as well.
It's like building a platform almost that makes the development of these personalized medicines accessible.
If you think about it, it's like the development of the personal computer. You know, the same thing. You'll have that. All of the computers were centralized before, you know, 1974, and then Apple came along and they said, "Here's the personal computer." And you think about the revolution that did. Obviously, we're not going to touch it because personal computer, every single human in the world will find a use for it. But we're talking about biological use. So cut that market to the biological use, and that's our market.
Can you big.
Yeah, it is
still very big, and probably, you know, bigger margins of course because of it. But
can you imagine 10 years in the future, like you're gonna have personal computers, personal AI assistants, personal medicines? It's gonna be incredible. We're just gonna turn into potatoes on the couch. We're not gonna do anything for ourselves. We'll be fed through a tube. We'll be thinking virtually. We'll be acting and behaving virtually in some type of metaverse.
Yeah.
I was even watching a video yesterday with you know those sort of seats from WALL-E? Yeah. Someone's made them. Someone has created like basically like replicas of seats from WALL-E where you just kind of lean in the direction of where you want to go, and it just takes you
I think I saw that video.
Yeah.
It's a prototype of the machine. Yeah.
It's incredible. Reality converges on science fiction and animation.
Well, I think it really does. That's actually a really good point.
So I think one more probably thing to add to that, obviously, because it's such an important and profound technology that if you can develop, I think accessibility is so empowering, but also comes with a significant responsibility because you're also talking about safety. You know, there's people could do things bad with this, and I'm thinking a lot about that too, because you can't just do one over the other. Same conversation is happening now with artificial intelligence. It's probably the same thing if you can create mRNA on the spot. So how can we safeguard, or mRNA or DNA, any DNA on the spot? So how can we safeguard against that 0.0001% of risk? But we know it's an important risk. You know, so you need to build these systems that are really integrated and smart to know if something bad is happening or being made, so that actually it doesn't get made. Or doesn't get amplified. So we really need to build that. We need to integrate different technologies to bring that to bring that empowerment. If we really want to kind of have the upside, but little or not of this downside, we really need to think about it in a kind of more responsible way.
I suppose that's where policy comes in. Policy and unfortunately the bureaucracy in the science.
Yeah.
To make sure that things don't get carried away.
It reminds me like of normal printers, where if you try and print, like, let's say an Australian or a US dollar bill, it will actually refuse to. It will print like 3 yellow dots rather than printing the actual note. So you're kind of, I guess you're envisaging like a printer that's
Yeah.
distributed, that's for mRNA. You can code whatever you want, but that it almost has like a machine learning model inbuilt that
Right.
detects if the mRNA that's being produced has a potentially nefarious purpose. Like if you're printing SARS-CoV-2.
Yeah, like that would be questions. In fact, there's regulations like in the US that are much better about that. I'm trying to argue for a regulation here, kind of part of few groups, but I'm gonna push them to do that. I think that's gonna be very important when these technologies become accessible. And let's get to it now. Let's start talking about it now, so nothing get rushed, because regulation and policy, if you get it wrong, you can really harm innovation. You really don't want to do that. You don't want to just regulate for the sake of regulation. You need to get it right, and it's important we do that now before these become available and possible.
Absolutely.
Who is important, or who is responsible for that form of regulation in Australia? Is there a particular minister that you would probably talk to, or
it would be a body of individuals?
Yeah, so definitely a few bodies. I mean, I think if obviously that these things would be regulated in the upper house first, and then so it's regulated in the parliament. But I think I'm not sure really who would enforce things. I mean, you could think about Biosecurity Australia, which would enforce something like that. You'll have agencies, so you'll have probably something like that. You'll have like a small agencies which monitor and regulate this, and I think that's going to be an important part to it, but and enforce it if necessary. So I think the other part is that whatever company or startups develop this, they need to have a responsibility to develop this, in a safe way, and they should ask for help. Like you can ask the government, we need this funding, to try to help us so we can regulate this. I think that's also startups are not doing very well at that, because that's a sort of nice way. They want to listen to that. They actually want to work with you, because that's sort of thing they love to fund as well.
Absolutely. Harry, I want to kind of break down a concept of, a lot of these innovative change makers in the world, they had some kind of experience in their formative years of development. So you mentioned you grew up overseas.
Right.
How was that experience, and how do you think it affected your ambition?
Yeah, look, I mean, I've had a very, I think in a way, somewhat fortunate but very difficult as well. So we weren't necessarily poor, we weren't that wealthy, but we were well off in Iraq. So I grew up in Iraq, and what happened is war came along, 2023, 24, and 2003, 2004, and that really just changed everything. And you really get the blanket get just pulled from you and just being thrown into this chaos and this hopelessness. And I think that's an important thing, because how can you build from that? How can you develop that? And then add to it all the all the security problems. So I think if you ask probably most Iraqis, we almost died at one point in time. So you would say goodbye to your parents. We used to do that. Like it's mesmerizing, but we'll tell them bye, and we don't know if we're gonna end up coming back or not. And that's the sort of thing we do this every day. And you'd have a lot of near misses. People try to assassinate us as well, because we were secular in a part where it was taken up by a terrorist organization. It was like one of those things. You just don't do anything. You just kind of, because you are, you can just get killed. So that was a very difficult time. But I think what I learned a lot about it is, and then what happened is that we moved about 2006, end of it, in Jordan. But effectively, we almost lost everything. All the house, we can't do anything about it, sitting there. We kind of really, we live in a good house. Now we just cramped in a small apartment in Jordan. We were then, obviously, we just thought that we can't come back, so we applied for immigration. That took a few years to materialize, to come here to Australia. But what happened did taught me a lot about surviving, knowing how to survive, and really aiming to thrive. And look, it's difficult. Sometimes it still gets me. I get back. But also, you really need to be just tenacious. You need to just kind of make things happen. I like to do that because sometimes you just have to walk was talking to someone about this today. You gotta have to walk through walls, because walls are always gonna be built in front of you, whether that's someone else is doing that, or the world is doing that, or it's just a metaphorical things happen, and you need to go through them. And that's the sort of thing you need to do, because otherwise you're not gonna be able to go anywhere. Yeah.
Prophetic. The walls are being built up for me as well in the research world. It reminds me a lot about your experience of war. Reminds me a lot about sort of my mother going through a similar experience. Like when she would say goodbye to her father. My grandfather was the police chief in Tehran.
Yeah.
So he'd go into the city, and she wouldn't know if he'd ever come back because there was so much violence at the time, both in the Iran-Iraq War and then later in the Islamic Revolution of 1979. I think it's just like it's almost like you actually experienced the war. Like you were kind of like the version of me that stayed in Iran, that had to go through all of that hardship. And I think hardship really does change the way you look at the world. It sort of builds resilience in you from the get-go. And obviously you've leveraged that, and you've built a name for yourself here as like one of the premier mRNA researchers. If you had to like go back in time though, maybe when you were closer to a younger kid, like what were you like? What were you like as a child? How did you see the world? What did your parents say about you?
I suppose as well, did that change over time, and how so?
Yeah, that's a great point, and I haven't really gave it a lot of thought, because I just haven't had a break to think about this for a long time.
He's like, I've been working since I was 4.
No gap.
Yeah, look, it is. I think we always think about that. Behind all this, there's a lot of glamour, even about me in the media and things like that. I can search my name, and I'm everywhere, but there's a lot of struggle, and there's a lot of moments of doubts, and but also I think what remains. So that comes through the war, actually, because that the war kind of or very, I would say, the big challenges that you will have in your life, they will give you positives and negatives. They will give you the resilience. They will give you, but they also gives you a lot of other stuff that I want to say shit. Well, so I said it anyway. Stuff that are that stays with you, and they harm you, and they really not great, and you need to really start understanding and separating those. But I think what it also get carried out is my enthusiasm, my curiosity. I was very curious. I love doing. I used to just kind of create things. I love to create things and build things, and with Legos or whatever I can find, and that really stayed. And also my enthusiasm that wasn't really surrounded around science, but it was just kind of around other things that I just love to do. And I think that kind of I used to bring things together, or bring people together, and make situations, or make kind of conversation. And I love doing that, because this is kind of the resourcefulness that kind of continued with me, and I think that stayed with me. And the love of creating things. I think that's when I was a kid. Yeah, so I was actually quiet. I wasn't that. I wasn't making a lot of mess. I was kind of good students at times and bad students at times, and still don't understand why. Just depending on my mood.
Yeah.
I feel like it's a dynamic thing.
Yeah.
Changes on circumstance and experience. You may go through a period where things are good, then you go into the trenches and things feel bad.
Yeah.
But then things get better slightly. So
Yeah.
I suppose throughout this whole... Sorry, you go. Yeah.
Yeah, no, I want to touch on that, because I think that's important. What is really important is that you just don't say, I am like that anymore. It's just you know that's a phase, and you gotta accept that, today and this past 3 months, you suck, and then that's it. It is just the way it is, but you need to see that you can come back from it. I think that's so important, and that's the resilient part. It's not really the failing. I think it's actually, I think there's a better term to it that I absolutely love, called anti-fragility. It's I don't know if you guys heard about that.
No, please explain.
Oh, that is amazing. I think that's the sort of things that I am trying to build. I think is very important. But so anti-fragility is a term that's brought by Nassim Nicholas Taleb, talking about systems, and I think he does mention people. I think he wrote a book about it. It's an important concept in systems, and especially financial systems. But you guys can read about it. So you can buy the book.
It's called Anti-Fragile, right?
Anti-Fragile.
Yeah.
Yeah.
I have heard of it.
Yeah. So it's so the idea behind it is that you have fragility. So the system gets stressed, it will break, right? Well, resilience, if the system gets stressed, you might kind of push it a little bit, but it just maintains what it is. But anti-fragility is when the system gets stressed, it improves and it becomes stronger. So it's like almost the stress is a positive indicator of it. In fact, if you create a system of anti-fragility for yourself or for a startup or something, what you seek is the stressors, is the challenges, is the problems, because you will be better at them.
Yeah. I suppose it's almost a precursor to a lot of success in people that you see online or even in the media. So it's an interesting concept, I think. So Harry, during this transformation of who you were and who you've become, do you think that you've developed a life's purpose?
Yeah, I think I'm not sure about that. I mean, I think that definitely needs a lot more thinking. I would say something about that. I think I am developing a mission, and I think missions are a bit shorter than life purposes, because I think it shouldn't be a mission of 1 year. It should be at least something around 5-10 years you do. So, you start with something, you start a few years. I think that's a lot more joy by the end of it. So my mission to develop delivery technologies for nucleic acid, for DNA, kind of evolved and gone into becoming mRNA. And I finished that. I've built the team, built the capacity for that mission. And I don't see, and my life has changed, and I grew up that I'm not what I started in 2010, I don't see it now. But that's about 13 years mission. Now I have a different mission that I can see maybe 10, 15, 20 years. That's a different one, which is enabling and empowering through by developing what we call that biocompiler. That's a sort of empowerment to develop tools that other scientists and medicine can use it. Doctors can use it. I think purpose is longer. I think it will eventually, by the end of it, you'll kind of look back at it. I don't think you can project it, but I do feel few things. I think one of them is what drives me is a sense of duty. I think that's what it really is for me. Is I have a duty to whatever the mission or the thing that I'm working on. It's almost like a civic duty. It's not like a material duty. It's a civic duty to give back to the world that has given us so much. If you look around, like all these amazing things, the technology that we have, is the sort of contribution. It's our duty, and actually our duty comes from engaging and deploying our experiences and skills. That's why it's dynamic. Duty is dynamic, because whatever you've learned, whether your skills, but also your experiences, the pain that you had, you want to formulate it and push it back. So if I'm a better entrepreneur than I'm a scientist, my duty is to go and do this, even though I may not like that to do that necessarily. I do love it actually, but it's an example. Yeah. So if I have the skills to be a scientist, to be a researcher, a typical researcher of what is that required, then it's my duty to do and go and do it. So, and part of it as well, part of duty is passion, but I think duty has that kind of components, and I think that makes you love what you do. I think that's kind of, you take it with you.
Yeah.
If you have the duty to change the world, then you are going to change the world.
Well, I don't think it like that.
You have to.
Yeah.
Right?
Yeah. I mean,
it's the intention and definition of duty. You do it despite how you feel.
Yeah, actually, that is a good definition. I think it's good to do something that you're passionate about, but that's the second part of it. I think that's the purpose, actually, also a purpose to live every day passionately, to do something that you love. Again, we live our lives every day. We don't want to just achieve things. we actually want to go to work and, spend the 6 hours, 7 hours, 8, 10, 100 hours, love doing something that we love. So I think that's another part of it. So it's not just duty, it's duty and, I guess, people say you have to choose between duty and love. But I guess actually sometimes you can do both. And if you do that, go there. Doesn't matter money, because it will happen. Money will come along, success will come along, fame will come along. Just if you find passion and duty, combine them in that Venn diagram and just find that common ground. And you say, I love entrepreneurship, but I love doing this part of it, but I also love and you can just do it and build that. Oh, it's amazing. Yeah.
That's fantastic.
Yeah. I think we'll move on to the quick fire round, but sure. I have one more question. I think when you're trying to think about the future, we've already discussed how can we position ourselves for the future. Obviously, you've mentioned that there's almost like two problems that need to be solved for this mRNA revolution to truly bear fruit.
Yeah.
One is scalable, low-cost, custom mRNA synthesis, sort of this bio designer thing that you are envisaging.
Yeah.
That's probably what your startup is related to. And then there's also the problem of delivery. How do we ensure that this is being delivered to the right cells, it's not causing toxicity, it's not causing adverse side effects, it's not like having off-target effects. Once those two issues are solved, once we have scalable mRNA and we have delivery that's specific, what is the next frontier? What is the future of medicine beyond that, in your opinion?
Look, it's a very difficult to predict, and it's if I can't see it that well, I'm not sure if I should predict it. But I can tell you there are trends, like we talked about, personalized treatment. A lot of the medicine going to be personalized. One of the things about there are going to be always new challenges and new combinations. So if you think about AI, for example, it's just an algorithm. It's in a way it's a algorithm on super steroids, maybe more. But the algorithm existed for about more than 1,000 years. So, but each time they're kind of been developed in a different way, and then you have been deployed to search, and now you have that advance. So it's very hard to predict. I think, although in biology is probably is easier to do, I do think that things like mRNA and genetic engineering going to be so prevalent in the world, because we have to meet the challenges that it's ahead of us. Things like the medicines, I think basically what's going to happen is some kind of quick revolutions and the cures. I think that's going to be something just like people not realizing how important it is, the CRISPR-Cas and mRNA technology to curing diseases, genetic diseases. I think that's going to just hit something, and there's just a realization how important that is, because you're talking about cures. This is not. This is the level, and I've seen them, and I can see them. I can also see them technologically happening. So you're talking about almost. So when the delivery is solved, for example, for liver, every genetic disease in the liver is within reach. That's how impactful it is. That becomes just instantaneously everything you can solve. You can develop these things for. Some of them better than others, but almost everything is. And that's incredible, because you'll probably just solve a lot of genetic diseases. People will not gonna have that. Just incredible.
Definitely. So we'll transition now into a bit of a fun area.
Okay.
It's called the quick fire round. Essentially, a list of AI generated questions, and your responsibility is to answer viscerally without thinking too much about the question. And they're usually quite ridiculous. So
okay.
Please don't hold us accountable for the quality of the questions.
Fire up.
So the first question is, what is your favorite place to visit?
Park.
Okay. What's your biggest pet peeve, or like ick? Something that annoys you, disgusts you?
Interruption.
Do you think it's possible to bring back a mammoth?
Yes.
What's
How?
your yeah?
Maybe
is the
how?
next
Yeah, maybe
question.
how.
How?
I've seen like a mammoth meatball, but I haven't seen a mammoth.
Oh yeah, I've seen that.
Should bring that on,
I
actually.
met the
we
people
probably
actually.
could. Yeah, we probably could get them on.
Yeah, they're in Brunswick. Yeah, the top people. Yeah, mammoth. I think there's a lot of innovations, obviously, in cloning. Even if it doesn't work in elephants, then there's a lot of innovations that I'm very excited about in artificial womb, artificial systems. They're almost hit it from the bottom and from the top. They're getting better and better from both sides. And there's a critical point where they need to do that. When that is kind of emerged, you'll have a basically you probably need another episode for that, not with me, with someone else, to talk about artificial womb, because that
Black
brings
Mirror episode.
brings a lot of the kind of sci-fi movies where you just have
a lot of ethics.
a lot of ethics on that. So, but obviously has amazing applications for people who for productivity, which is another massive problem that we have in the world. Yeah.
What's your favorite TV show?
Favorite show of maybe How I Met Your Mother's Community. Actually, Community. I would say Community.
And why do you like it so much?
It is so good. It's just nerd, my sort of nerdiness. I love it. It's Dan Harmon.
Great. What's your favorite fast food?
Pizza.
Solid.
Again.
Imagine your house is on fire. After saving your loved ones, you can only save one item. What would it be and why?
Don't think. Okay.
Prototype to the bioincubator thing.
Maybe. What house? What do I have in my house? Maybe I have a set of Lego that I brought when I came. Yeah, it's meaningless, but I like it. Yeah, because it's sentimental.
That's a good one.
Yeah.
That's a good answer. So the last one, maybe. Yeah.
Maybe
a couple more. Okay. So, what in your life do you feel you're most grateful for?
Look, support of my family, some of the people that I work with, especially my supervisors, support of my family partners. I'm really grateful for that, even though I need to probably do more. We all probably are, because we're here. But I'm very, very grateful for that. Grateful being here, grateful being alive. At one time I didn't think it was gonna happen, but I'm here. So grateful to be here.
That was deep.
Wow. What's one subject that you'd like to learn more about?
Oh, enzyme engineering.
Yeah.
We were talking a lot about that. I think that's an important thing. That's the sort of maybe scientific thing. I would love to learn a lot more about safety and policy. I think that's the sort of thing that I also want to learn. History, I am already doing something about that, so I love history. That's essential. But I'm already trying to learn a lot about that. But so a few things like.
Yeah, that kind of goes into another question, which is, if you could go back in time and redo it, what would you change?
I think I would have maybe started earlier on what I'm doing now, or what I'm trying to do now. I think, it's just kind of losing that doubt and understanding how when you put your head to something, it actually works. It's like a magic, but it does. Because one of the things this is my thought about it, and I probably would finish maybe something for you guys, it's actually how we think about risk is phenomenally flawed. And I've seen it firsthand, and I thought so much about it during COVID-19. But risk is a dependent variable. It's not independent. So when you say 95% of success, that's on day one. But when you build on day two, that's 94%. It's a dependent variable in statistics. So every time you solve something, the risk goes down a lot more. So you should not be afraid to do things, what I mean, because they have a 95% success. So it's like a very important that the risk, you can change it. It's not really written.
It's not fixed.
It's not fixed.
Yeah. It's a good mental shift to think about whether or not you should pursue something.
Yeah.
It's not as risky as you probably think it is.
Yeah,
you can change the risk over time.
absolutely. I mean, I think obviously there's some things that will just kill you, and you're never gonna get 100% success, or like 100% or 0% risk. You're always gonna get like 30, 40. Like you'll always have that, but it's not 95. That you can change, and you build every time you achieve something that goes down.
That's a good point.
So when you achieve every milestone, whatever you call it, something you achieve it, then that clicks, and then the risk of something not succeeding goes down. I did not thought about it. I think that's so important that I wanted to, if I could have known, I wanted to have learned that really early on.
Well, you've taught it to us young.
Now you do.
I'll take the lesson.
You apply it with it.
Last question. Actually, I'll do one more question from Oliver as well.
Sure.
What is a bad habit that you wish you could get rid of?
Yeah. I mean, I think I'll probably just say this generally, not like necessarily silly small habit. I would say lack of focus. That was something that I had, and I had fought very hard, and I think I've successfully fought, because it was one of those things where if you have the habit of focus, and I'm not talking about going in during the day, opening and that's important because you need to get things done. I'm talking about that long-term focus. The habit, I think that the habit of me saying yes to things, a lot of things, everyone, even especially if you're an entrepreneur, you'll love doing that, because this comes as a package that makes you successful, actually. And it's why it's so challenging and why we have to fight so hard, because it's the single most important thing, I think, in being successful or being anything is focus. It's absolutely, I mean, it's one of those things where you have good advice. This is kind of great advice, and then I'll give you something on top of it. Focus is so valuable. And it's hard to do. It's so hard. It's an everyday thing. I haven't got there yet. I'm gonna have to have a long time to go, but focus.
Yeah, I feel like it seems sometimes people get disgruntled when things don't go as they plan, and they lose focus because you almost go through this cyclical curve of things are bad now, so I'm gonna look for something new. And then you've got like that uninformed optimism. And then you go into that, that pit of informed pessimism because you're like, wow, this is way harder than I thought. And then you cycle back to the uninformed optimism. So it's almost that valley of death, they call it, right?
And there's something saying, without taking this for too long, but, Steve Jobs said this. He talks a lot about focus, and you can find that. But there's something about it he said, and that stuck with me, just revolutionized my head. He's like, I'm proud of the things. He's following up on how focus is important. He says, we're proud of the things that we didn't do as much as we're proud of the things that we did. It's just so much weight on saying no, on the things that he said no to. He's like, I'm proud of the things that I said no to. So it's like one of those things where like, I understand that. This is why focus is so important. But anyway, I think saying yes to things it's very difficult. You have to be very calculating. You have to have fun. You have to have a margin of saying yes to things. But you're just going to be very, careful.
We're probably going through a bit of a phase right now where we're saying yes to too many things.
Yeah, learn it. Yeah.
Obviously, there's a stage in your life where you've got breadth. You're trying to get experience, and then I think long-term success really comes at the cost of singularly doing one thing at the exclusion of all else, but having picked that one thing very carefully and very deliberately with enough generalist experience of the space to minimize your risk as much as possible.
That is so important. There's actually someone written about it in Nature paper called Explore and Exploit. It's a theoretical phenomenon. So he actually has modeled most successful scientist in across all fields. He used language models to define that, and he did form a theory called explore and exploit. It's so important, but it's just like that. It's explore doesn't work. Exploit doesn't work. exploit then explore. You have to explore then exploit. It's just one of those things where you broaden first, then you pick something and you just fight. But it's weird because you learn all these skills and you build all your personality and your habits, and then shove them out the window and build new ones. But that takes time, and we just have to be okay with it. Yeah.
And last question, what's a time that you've laughed uncontrollably?
What a time that I laughed uncontrollably. Yeah, hanging out with my friends. Maybe like you guys, you gotta laugh a lot. Yeah. I love doing that because you just kind of, I think making fun of ourselves. I remember having a party, so we just couldn't stop laughing.
I think there was a time when I was going to a physics class and I just started laughing and I don't know why. And I actually couldn't contain my laughter. I was sent out of the classroom. Thought I was having a stroke or something.
I think it's those moments are, they don't come by, so often, but they're incredible when they happen because so many endorphins, so much, adrenaline and such that you kind of forget about everything else in the future for a moment, and you're just stuck in the here and now, which is quite a nice feeling sometimes.
We'll end with that, right? Here and now.
There's one last question that we have for you, just to end things up. If your life were to be summarized into a single mission statement, what would it say?
A single mission statement. I don't know. I mean, I think it's a dynamic, so I'm not sure if sorry for being a bit of grump about it. I mean, I think, betterment and advancement of society through service and duty. If you think like my own personal mission, I don't know. I think that also ties very well to the startup mission that we're setting up. You do have to have that. You're part of it, especially at the beginning. I think that's it. You really need to, service is important because that empowerment. You give people, help them to help themselves. This is really important. We've done it. I'm doing it now. This is actually the major form of what we do at our lab as well, because collaborate a lot with scientists who have all these diseases and things like that, and we help them by sharing the technology.
Yeah, I think that's
more than sufficient. Yeah,
that's more than sufficient, and I feel a good amalgam of kind of your story that you've described on the podcast. So we appreciate you. Yeah, we appreciate you giving that perspective. Yeah. One last thing before we just wrap it up is a question that you would like to leave on Orange for the next guest.
Great. Next guest. Well, without knowing what the next guest would do, I think, it would be great to know, especially I'm sure you guys getting great people here. What sort of, what sort of mental model they use to kind of that drives them. I guess, like you touched on that, the mission. I think that would be nice to learn a lot about that, what they've learned, their history. Always love hearing about that.
Yeah, it's a good question.
Well, we'll leave it at that.
Absolutely.
Thank you, Harry, for
my
coming
pleasure.
on.
Thank you for having me.
This has been an episode of the Orange Podcast, and we appreciate you coming on.
Absolutely.
Same here. My pleasure. Thanks, you guys.
Harry.
Now, we talk a lot about business and entrepreneurship, but to build the future, you need people and a place to build from.
Discover the perfect workspace for collaboration and productivity.
Melbourne Connect Co-working is offering our viewers an exclusive 25% discount on 12-month memberships.
Quote Orange when you submit your inquiry. Links are available in the description now.